And I got this thing recording, so we're ready to go. So while I'm doing this recording, as I always do, I'm looking at the exam. Did anybody have any specific questions about anything as you were reviewing? Okay, so you weren't just doing the general review thing. That's what I figured. I mean, it's sort of reiterating it. And I had done that. I guess the one I sent you, I had done previously, and I don't think I changed the exam from one of my previous ones that were there. It starts with basic nerve anatomy. Again, receiving end, typical dendrite, transmitting end, the axon, multiple inputs, multiple outputs. We use the term cell body. I think that's the term I use mostly. There's a couple of names. Remember, if I use the term nucleus, it's a cluster of cell bodies in the central nervous system. If I use the term ganglia, it's a cluster of those cell bodies outside in the peripheral nervous system. That's a significant difference that's there. So, again, the big deal, they come together, it's called a synapse. I just use the term, there's a lot of synaptic cleft and all that stuff. For me, I like to keep it relatively simple. And the most common junction you're going to see is an axon and a dendrite, by far. That's really the one I'm referring to without going bananas over that stuff. And so the receiving end is the dendrite, the transmitting part is the axon. I mean, it's relatively a simple connection. The simplest connection by far and away is a reflex, okay? Knee-jerk reflex. We used to think it was just sensory and motor, knee-jerk, boom. But the reality is we know there's something in between called an interneuron, because somebody whacking your knee. At least the message is going somewhere to your brain. Hey, it's a knee-jerk test. It's very interesting, but that's the simplest circuit. It's a 3-nerve circuit. We know that today. A sensory input of some type, an in-between or a connector, and a motor output. That's as simple as it gets. Most circuits are immensely more complicated. Reflexes by far and away are the simplest circuit. I spend a lot of time on these glial cells, and so there's a number of questions about it. And by definition, they're connective tissue, and they're the connective tissue that's dedicated to the nervous system. That's really what their role is, type there. So whenever you hear when we talk about pure nervous activity, things like threshold and action potentials and all that type of stuff, those are the nerve-to-nerve communications. The glial cells just basically support them, and whether it's an astrocyte that's by far and away the most abundant and effectively protects them, guides them, nourishes them, the whole thing, it's an intricate part of the blood-brain barrier. You remember the blood-brain barrier is mostly from the unique capillaries. You get more capillaries when you get to AP2. Capillaries have so many tight junctions that water-soluble materials can't effectively get through. And on top of that, it has the astrocytes, these glial cells, and literally through the glial cells, which we'll go back and look at that illustration of the PowerPoint, it embraces the nerves themselves and it embraces the capillaries. And so those materials that are fat soluble and small enough, or things like oxygen and carbon dioxide, literally are able to get through the membrane of the capillary, travel through the glial cell itself along the membrane, and get into the nerve cell through the membrane, because they're all soluble. They're all by definition small, non-polar, fat soluble things. So it's about the size and it's about the solubility. So non-polar, fat soluble, simple gases like that, which always are, particularly oxygen and carbon dioxide, are in that. So they're really a significant part about it. And like I touched on, threshold, and all that means when you hear the term threshold, if you got the threshold, you get an action potential. We don't have end plate potentials anymore because we're not looking at muscles. The potentials we have, really fancy terms, really they're either excitatory, things that have a tendency to depolarize, bringing it closer to threshold, or inhibitory, bringing it hyperpolarizing or bringing it farther away from threshold. So we really, you have a slightly different terminology, because unlike the muscle system, depending on the neurotransmitter and the circuit that's involved, it's either going to be make it lower, it's either going to excite or inhibit, more likely to fire or less likely to fire. And we sum those all up. And if we get, and whatever the summing is, if we get enough to reach threshold. Remember, it's an all or none phenomena, and it's a unidirectional phenomena. All big differences from muscle. Muscle goes everywhere throughout that muscle fiber. Here, from wherever it's stimulated, down the axon toward the end. That's the electrical traffic. Again, the interior, they're moving neurotransmitters in that direction, but they're moving them back to reprocess and blah, 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 all that stuff. So there's two-way traffic in the nerve, but not when it comes to the action potential. That impulse is only going to go from start to finish. Typically, it starts at that area, that interesting area called the axon hillock. Let's see. So, oh yeah, I forgot. So, and the other thing, when you have those cell bodies all together in the central nervous system, typically cell bodies, synapses, and dendrites make up what we call gray matter. More than anything else, everything else is principally white matter, and white matter is more abundant because the axons are longer, and inevitably, there's just a large amount. There's so much communication, it has to be rapid, and that's where the white matter comes in. And remember the distinctions in the white matter. Up and down projection, going across commissural fibers, right to left, left to right, and then association, same side fibers, basically processing the information and shuttling to where it's supposed to be. I'll go back to some of this. I'm trying to keep it sort of organized. I'll go back to some of this stuff. Okay, but you do, what I'm seeing is a question that comes early on. It basically says, remember the four nerves from the parasympathetic, four cranial nerves. It's a little out of sequence. Just somewhere write down. When you get here, it's open book, 3, 7, 9, and 10. Beg your pardon, 3, 7, no, I got it right. It's okay. So I'll get back, and these are like the fight or flight stuff and blah, 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 blah. I'll get back when I get back there and take a look at this. Important areas, the other things I want to get to the glial cells part. So the glial cells, again, ones that are part of the central nervous system, astrocytes, oligodendrocytes, ependymal cells, okay, and those microglia. Okay, the astrocytes talked about. The oligodendrocytes, they provide the myelin sheath. The unique thing about them is compared to Schwann cells in the peripheral nervous system, Schwann cells wrap around those very long axons at intervals, obviously, leaving those nodes or sheath gaps. Oligodendrocytes do the same thing because there's such a space limitation. One oligodendrocyte might be effectively doing that chore along axons of maybe up to 60 different nerves inside the brain. So it's a much more compact system, would be the best way to describe it. Obviously, they're thinner, it's a much more compact system. That's an important part of it as well. Microglia are basically little scavenger cells that provoke, they're almost like traveling white blood cells, but they're really gobbling up debris, and so they're acting almost like an immunological function that's in there. And the ependymal cells line those areas, the choroid plexes, where the cerebrospinal fluid is kind of finalized, and the fact that they have cilia, the beating of that cilia creates sort of the flow of the cerebrospinal fluid. So again, the blood-brain barrier, capillaries, tight junctions, and is surrounded by glial cells. Just talked about the cerebrospinal fluid. Okay, it's basically more or less a filtrate, okay, from the blood, but it is modified in some way, shape, or form to make it, by the action of the choroid villi, and then moved around by the ependymal cells to be different. So it's really a filtrate with some modifications. Okay, so if you wanted to summarize it in a good way, it's produced in the central nervous system. It's not in the bloodstream, but it has a lot of similar components. That might be the best. If you were going to make a generic, sort of a decent description. more or less generically. Oh, I'm a nut for ventricles. Okay, and really they're one continuous hollow fluid chamber, but okay, you have again, what are the ventricles associated with? The cerebral area, the cerebrum, the lateral ventricles, the same. They're C-shaped, they're large, and they kind of follow the famous lobes, the frontal, occipital, okay, temporal, okay, parietal lobes, all of that in that area. Then the ones that are around the diencephalon are the third ventricle, because it used to be first and second, now they just use lateral, same on both sides, lateral, lateral. Third, around the diencephalon is the thalamus, the hypothalamus, and the epithalamus. And then there's an aqueduct, basically a fluid passageway that goes through the midbrain, and it opens up behind the between the cerebellum and the pons and the medulla oblongata. You have the fourth ventricle. So important because that's where the egress passages are. They're called apertures, which just means an opening. Two laterally, one median or midline. And that's how the cerebrospinal fluid escapes from the ventricles, not all of it certainly, and enters the subarachnoid space. Then the ventricles, after the fourth ventricle becomes a central canal, kind of started at sort of the base of the brainstem in the medulla, and that really tiny, almost pinpoint passageway all the way to the bottom of the spinal cord, and then it can exit there and fill the subarachnoid space. So again, fluid inside, fluid outside, buoyancy, protection, bathing it, electrolyte balances, da da da. That's what. So it's a very unique fluid in that regard that are there. Let's see. Okay. Big player in the commissural tracts, corpus callosum. Got to know that. That's that big, thick white matter that goes side to side. See, we don't have the luxury here. It would be nice if we, in a lot of schools, whoever's doing your lecture does your lab. We see a lot of that, like in community colleges. You have lecture and then you walk into the lab with the same person, so you can sort of reinforce the ideas. But when we have have to have 6 or 7 labs, it's a little hard to do that type of thing. I think lab will go easier for you folks, AP too, because you'll have Dr. Robbins won't have to accelerate his class. You won't have to listen to me. Could you smile when it hurts? Thank you. You're not a Steeler fan? You don't care. Oh, I've managed to get my wife to become a sport because she knows her dad, she'd never be able to talk to me. What we need to do is don't talk to me at the game's up. Do that now after the game's over. Okay, we did that part. Keep going. You're in there. Did the ventricle. I'm going the wrong way. Got that done. Okay. A factoid. What I call factoids. A little bit of extra information about each one of these systems is appropriate. So, for instance, the frontal lobe, cognition, being awake, directing muscles to move, reasoning, speaking, all of those kinds of things, communications, all the advanced processing things, and generating to effect, impacting things like that. So conscious control of motor activity of any way, shape, or form, whether it's speaking, acting, physically moving something, that's the frontal lobe. General sensation, parietal. Okay. Visual processing, and a tremendous amount of circuitry in there, occipital lobe. Hearing and balance, temporal lobe. It's very generic, but it's important. Those kinds of things. When you get to the diencephalon, the big player with that is the thalamus. Effectively, that's a relay station. It does a lot of things. I mean, it's subconscious. So, I mean, the way you describe it from a book perspective, it sorts, edits, and relays. Basically, pre-processes information, almost assigning an importance to it, and basically putting it to the right part of the brain. Oh, this sensation you better pay attention to because it could be significant. This one, you just file it in your memory bank. At the same time, it's like me. I'm talking to you, all of a sudden I see someone walks into class. Well, I know they're walking into class, but it's not a big deal. Or they're walking out. Not a big deal. So sorts, edits, and relays, sends it to the appropriate part of the sensory aspect of the brain, not directly to the motor. So it's parietal, occipital, and temporal. are the lows. And the oddball one is the insula. That's very primitive. That's typically taste and smell, but it's really important because the primitive nature is why, particularly with smell, why smelling salts work. It's part of that is it keeps you awake and alert, those types of things, and that's that reticular activating system. That one that basically we call it arousal, but not arousal in a sexual sense, just arousal, getting your alarm clock going, that type of thing. If I'm lucky, somebody, my wife got up early and made coffee, so I'm something like, that's good. Which never happens. I get up like 5 o'clock every. You don't even want to know me. The hypothalamus is really where the blood-brain barrier is weakest. That has a tremendous amount of sensors because of that, whether it's sugar, salts, all those types of things. It also plays a pivotal role in emotions. It's part of that limbic system, the emotional system. What's neat about that, it has a tiny stalk that protrudes downward and really becomes what we call the posterior pituitary. And you'll hear about that when you do endocrine. And the glandular portion or anterior pituitary is adherent to it. So that's the juncture point between the nervous system and the endocrine system. And that's because a lot of times, where again, nervous system makes rapid changes, it influences the endocrine system to release hormones that make the changes last a little bit longer. Again, things that happen fast go away fast. And by endocrine, it's by no means slow, it's just slower. So where this may be fractions of a second in nerves, in the endocrine system, it can be seconds, minutes, hours, days, whatever, weeks. There's the timing. Long is a very non-specific term, or longer that's there. And the little stalk has a fancy name. It's called the infundibulum. And I made a big point, and this is the reason to remember it. When we start to get what the other area in the cerebral cortex that we don't talk about quite as much is called the basal ganglia or basal nuclei. Either term used, because in the old days we called them the ganglia. They are a unique system that has a lot to do with motor activity. It's really sort of the finer aspects of controlling motor activity. So generically, we talk about motor disorders like Parkinson's. That's the big area that's associated with. There's a tract that goes from the next area of the brain called the midbrain, because that's where neurotransmitters are mostly produced, the major ones. Dopamine, norepinephrine, probably some serotonin. They're all made in that midbrain. That's why it has those pigmented areas like the substantia nigra and the red nucleus, because typically that's a sign of neurotransmitter production. There's a pathway that goes from the midbrain to the basal ganglia. That's the one that's implicated in Parkinson's. It's because dopamine is not being produced sufficiently, and we utilize that idea. There's another pathway that goes to the limbic system. That's the one that's associated with addictive disease. Like when I do the physiology of this in my advanced class, we get more into those types of things that are there. And so we talk a lot about it, and we're just starting to touch on the neurotransmitters. The neurotransmitters are a big player. And again, this is the same kind of question you saw with muscles. How do we get rid of a neurotransmitter? It diffuses. And in the central nervous system, we do a lot of reuptake. Okay, and the other thing, it just breaks down metabolism. So you have some type of enzyme like acetylcholinesterase, other ones that it was not necessary for me to name, that break those substances down. Reuptake is a big play. There's a lot of recycling, and particularly for serotonin. Remember, I commented on those. We have serotonin is very selective. It's about the receptors. And all of those medications that we use constantly these days for mild depression, eating disorders, ADD, many of them are really based on serotonin. The fancy name is SSRI. I wrote that up on the board, selective serotonin reuptake inhibitors. So serotonin is a big player with regard for that. And of course, the one that's most important to know about, because it's everywhere. It's in the brain, it's in the motor end plate, it's in the autonomic nervous system, it's acetylcholine. Thankfully, it's the first one we get, and it's the one that we need to know the most about. The meninges, or meninges, however you want to pronounce it, doesn't matter. Three layers. Outer layer is dura, strong. Inner layer is pia, delicate. And then that arachnoid layer, because it has those strands that kind of look like a spider web, and it's between the cerebrospinal fluid is between the pia, the deepest layer that's adherent, and the arachnoid. And again, the big deal, the distinction between brain and spinal cord. In the brain area, the dura has certain areas where it splits to allow the cerebrospinal fluid to join veins that are in the blood vessels, and effectively they're mostly veins. In those splits, they're called sinuses, flat areas that are wider, but basically are sort of widened and flatter veins. And it's in those areas where there's little overflow valves. Okay. They have a fancy name. They're called arachnoid granulations, and basically they're like an overflow valve. So basically, the cerebrospinal fluid has an escape path to come back and join the blood going back to the heart, and everything gets recycled. And we make about a pint of that a day. Roughly a half. I would say it's about a pint of that, rather not a half pint. It's about 500 milliliters a day, and we absorb about 500 milliliters a day. That's the whole idea of balance. When there's imbalances, of course, there's a lot of problems that are there. Ventricles. Oh, you got to know what the arbor vitae is. That's the cerebellum. That's that distinctive tract. You should have seen that on the dissection. I would think you did on that. Very distinctive because it was such an interesting structure. That really is because the cerebellum, remember what the cerebellum does? It's basically coordinating what they call ipsilateral. Most of what happens in the right side to the left side to the right, we call contralateral. Works on the opposite side. The fibers cross over, the fancy term you should know, it's called decussate. The cerebellum is all same-sided. That's why it's ipsilateral. And so the definition is ipsilateral motor coordination. Effectively, it plays a big role in muscle memory. So much information. It's getting information from below. It's getting information from above. It shuttles the information forward, down, up. It's an extraordinarily intricate area. And so it has elaborate white matter tracks, and that one that has that interesting shape called the arbor vitae, or tree of life. Very famous term. I'll spare you the anecdote. That was the famous synagogue where the terrible murders occurred in Pittsburgh. Remember that? Oh, you gotta go look in your history. Tree of Life Synagogue, October I think it was October. I think it was 8 years. Maybe it was October of 2019. Crazy guy came in there and killed 11, 12. One of whom was my high school classmate, doctor. It's the same synagogue in Squirrel Hill that I got married in 1978. First one. Big deal, big deal. He's on death row. I'm going to stop here before I get angry. So you guys were pretty young when all that happened. Go back and check it out. Because it's such an important area locally. They just came out with an interesting documentary about it, where they're interviewing survivors and survivors' families. It's very, very interesting. There had a lot of ties, a lot of local ties to the community that were there, and because there were actually 3 synagogues on that Saturday morning in October, and I came out. We have my graduating class in high school. We have a scholarship fund, so I kind of like the guy here who keeps me informed of local. Moving on, so some anatomy is there as well. This is the longitudinal fissure. Okay, separates the right and left lobes that are there. You have that lateral sulcus that separates really the temporal lobe. You should remember a little bit of that. You should certainly remember the locations that are there. And then we get to the spinal cord anatomy. The big players are the ones I wrote on the board. Cauda equina, the horse's tail, all the nerves that basically the spinal nerves that exit below L2 go through. They continue to exit through all those openings, but the spinal cord terminates usually between L1 and L2. That conus medullaris, it comes to that conical ending. The filum terminale is that strand of connective tissue, with meningeal tissue surrounding it that anchors it all the way into the sacrum and the coccyx. So that's all those terms. Chorda equina are going to be on here. Phylum terminale is going to be on there. Conus medullaris is going to be on there. All of that stuff is part of it. Oh, and the other one, I knew I remembered it. The central sulcus. That's the one, you remember the diagrams, the homunculus, that funny looking up into upside down guy? Remember, you have the motor gyrus is directly anterior to it. That's the primary motor gyrus. It tells me to move something. Where we sense something, like idiot me falling and cutting my finger last week. Don't walk on wet leaves because you don't know how slippery it is. Rule number one. Don't be clumsy. Rule number two. And the one behind it is the primary sensory cortex. So motor is anterior to the central sulcus, sensory is posterior to the sensory sulcus. Again, if it's hyperpolarizing neurotransmitter, less likely to fire, farther away from threshold to polarizing closer. Okay, talked about that. This is good. Oh, again, the blood-brain barrier, I can't emphasize it. The things that cross non-polar, and the other thing that most of you should be, I don't know if I made a big point about it, alcohol. Alcohol is interesting because even though it dissolves in water, it still is non-polar enough to go through. I mean, it's like when somebody ingests alcohol, what happens? It goes to your head. None of you would know this. My eldest is, he takes care of the family's alcohol consumption. He's going to live for 2 years in an alcohol-free country. He's going to be in Bahrain for 2 years. On a military base, I don't think it's quite a naval base. I think it's a big deal. Okay, and then other neurotransmitters. Glycine and GABA are consistently inhibitory. Glutamate and aspartate are consistently excitatory. That's important. The catecholamines, by definition, dopamine and norepinephrine. So a lot of that's important. I'm not asking necessarily where they came from, just to amplify it. Dopamine and norepinephrine, because down the road, I don't think it's on here, but they come from tyrosine. Tyrosine's a big player as time goes on, as you will see. See, in that general anatomy of the cord, motor cells are anterior or ventral. Sensory, in the gray matter, sensory material is posterior. So the motor cells are generally located in the gray matter anterior, sensory in the gray matter posterior. I'm using those terms rather than ventral or dorsal. Oh, I did make the big deal about the lumbar punctures, the spinal centesis, that type of stuff. I think it's really important that the reason we're able to do that is the cord terminates. If we do it at L4 and L5, which is typically where we are, in the space in between there, the cord stops at L1 and L2. So again, you don't run the risk of sticking a needle into the middle of the cord and causing a spinal cord to be significantly damaged, because the nerves have much more give to them. They're not tethered like the cord is. So that's why it's done there. It terminates above the level of the puncture. Plus the anatomy is such that we don't have that double layer of dura. It's just surrounded by that fat. And the fat's significant because that's where we administer epidurals. Okay, and remember, frequency equals amplitude. That's very important. The hardest question is a sequence question. It's effectively a 5-point question. I mean, it should be helpful since you can look this stuff up. Because given that you're here, the sequence of events, effectively, what goes on, it's specific in a nerve. It's nervous system question. What happens? Some kind of, basically for a nerve to fire and to go reach an action potential, some type of neurotransmitter has to be fired. It has to work its way across and be an excitatory or depolarizing impetus, depolarizing nature, to bring it to threshold. Once threshold occurs. So the neurotransmitter goes across, it triggers the receptors to bring it closer to threshold, and once it reaches threshold, an action potential occurs. When the action potential occurs, it's going to means that that impulse has reached first the sodium and then the potassium voltage gated channels, and once they fire to reset the whole system, then the sodium potassium pumps are activated. So you get a neurotransmitter has to be released. It has to excite the area and excite the receptors sufficiently to reach threshold. Once they reach threshold, there's an action potential. In the nervous system, that means it's true in the muscle system, it's slightly different. It's going to trigger first sodium and then potassium voltage gated channels. That's going to set up that chain reaction of the impulse. They'll take it down and effectively fire the nerve. Back at the ranch, to constantly reset the system for the next impulse, the sodium potassium pumps become active. Sequence questions are hard. You're going to get used to them because you're going to see the same things. It's very funny. One of our professors, she had just a totally different discipline. She had passed a complicated exam to certify her to teach something in software engineering. She's brilliant, and she said, they give you this whole thing, and then they have a question with 4 answers, and they seem almost identical. And I looked at her and I said, "This sounds like a nursing test." You get 4 completely answers that all almost look identical with subtle differences. It's up to you to pick the difference. That's the complexity of the nursing exams. Well, I should bring my wife in as a guest speaker. That was very frustrating. You told us this in class. No, but that's not the answer I wanted. But you told us this in class. It's okay. Let's see. We covered that. I'm doing good. Now it's time. Parasympathetics and sympathetics, the autonomic nervous system. Again, it's all fight or flight. The one question about the anatomy, remember that if you have long preganglionic fibers, think about the vagus nerve, because that's really the big nerve in parasympathetics, and it has short postganglionic fibers. The sympathetic nerves is just the opposite. Effectively, all of them are so close to the spinal cord, they have short pres and long posts. So that's the idea. Somehow remember that. Again, not only is it open book, you sit here and write on the test. Short pre, long post, sympathetic, long pre, short post, parasympathetic. There'll be a question. Get it done. That's there. And then I have a lot of questions that say in fight or flight. Okay, what happens? What changes occur? Do your pupils dilating? Do you have increased blood flow to what particular area? Do you have decreased blood flow? So remember where basically what happens to these things. Got to alter that question. I'm glad I looked at it. Okay. So effectively, because I hadn't proofread the test, but I'll make, when you come in, you'll see I'll be making a subtle correction on the day that you folks come in and take the exam. Eyes wide open, pupils dilated, respiratory passages open wide. Those are the bronchioles that are there. Your respiration increases, your heart rate increases, your blood pressure increases. You redirect blood flow to your skeletal muscles, also to the heart, also to the liver, because that's where your energy supply is coming from. You not stop, but you really severely limit the blood flow to the gastrointestinal and the genitourinary structures. That's the big thing. And they have a lot of questions that are based on that. What do the pupils do? They dilate if it's fight or flight. If it's parasympathetic activity, they constrict. When the adrenal medulla releases epinephrine and norepinephrine, it comes from by way of the sympathetic nervous system. Oh, I forgot to make mention about myelin. Myelin is all about how thick the nerve is and how much myelin, that's what makes it faster. Okay. Almost there. Okay, remember the other thing, just to emphasize the point, the two diseases that we see associated with the basal nuclei, I'm kind of summing up things, are the are both Parkinson's and Huntington's disease. In the old days we called it Huntington's chorea, Chorea. They're both movement disorders, jitters, shakes, etc., like that. Those are both very much associated with the basal nuclei. Uh, Remember the big one that we saw the most of, the best example of sympathetics and parasympathetic systems working together, again, is the sexual response, both excitation and climax, orgasm, ejaculation, whatever way, and I frame it differently. Oh, and then, injuries, when you hear the term concussion versus contusion, a concussion implies temporary damage, be it to the brain or the spinal cord. How temporary is a pretty vague term. Contusions imply damage, permanent damage, so you've messed up something inside. That nice article in the paper about Ryan Shazier, the Steeler, who had that. I'm just checking to see if I missed anything. Just about done. Again, 3, 7, 9, 10. The vagus nerve is the 10th. That's the long one. That's the main one for parasympathetic. The only one leaves the head and neck area. It's there. I think that covered all those different areas. I think we're good. All right, we're good. We are good to go, everybody. So, please, any questions, just come in and ask.